So we're talking about pain again, but this time we'll be touching on bone metastasis, which is basically the spread of cancers to the bone. Now, this can cause severe pain, likely due to the significant amount of nerves and innervation you can find in the bone. And just remember that regardless of where the pain mediator actually binds with the nociceptor, that it can be found on the axon, on the cell body of the neuron in many different places. So when you have so many neurons present inside the bone, regardless of where that mediator is released, it's likely to find a neuron, and that can send the pain signal. So that might be a reason of all the severe pain you get from this type of condition. And it's characterized by two different types, or two different symptoms at least, that you most likely see. One is osteolytic bone metastasis, which is basically when there's a presence of cavities or increased bone resorption inside of your bones. Resorption basically means degradation. Or you can also have osteoblastic bone metastasis, which is basically again increased formation of bone and too much bone being formed. And this is not necessarily stronger because the new bone may not be as efficient or effective at actually strengthening your body. So you can still just step on it incorrect way and end up snapping your bone without even realizing it. This looks complicated, but I guarantee you it's much more simpler than you think. So today we're going to be looking at the glutamatergic neuron, and we'll briefly mention GABAergic neurons, but mostly glutamatergic. So here is where you're familiar with. You have your glutamate inside of your vesicles, and it's just released into the synapse. Now, the glutamate, only a very small portion of it actually binds with receptors in the postsynaptic membrane. This can be like NMDA receptors, something of the sorts. And that excites that neuron, right? You get cations flowing in, depolarizes the neuron. Now, what do you do with all the excess glutamate out here? Well, a very, very small portion might get reuptaken by the neuron, gets restored into these vesicles. However, the vast majority of it, around 80%, are taken up by these glial cells that surround the synapse called astrocytes. And it can be done through a channel such as the EAAT2. We didn't really mention it, but thought it'd be important to know. But these astrocytes are glial cells, and they basically do synaptic cleanup. So what happens is that most of the glutamate that's taken up is actually broken down by glutamate dehydrogenase into alpha-ketoglutarate. This happens in mitochondria, and it enters a Krebs cycle. The alpha-ketoglutarate is actually one of the species used in the Krebs cycle, and that's eventually used to make ATP. So most of your glutamate actually makes energy, but then a small portion of it comes back up into another part of the astrocyte where glutamine synthetase, this enzyme right here, breaks it down to glutamine, and that's eventually transported either to your glutamatergic neuron or to your GABAergic neuron. And then that's synthesized into glutamate through glutaminase, and then the whole process takes over again where it gets stored and can be released. Now on the other side, your GABAergic neuron also takes up that glutamine, converts it to glutamate via glutaminase. Ignore my typo. And then glutamate decarboxylase will convert glutamate into GABA. So this is actually one of the only enzymes that's different between the two neurons. And then everything else is basically the same for GABA, except it hyperpolarizes the postsynaptic neuron by opening anion channels. So this is a bone. Say there's a tumor cell here. Definitely not proportional, but tumors require and use a lot of energy, right? They have a high metabolic rate. Now, what this means is that you have tons of mitochondria that are overactive, and overactive mitochondria cause oxidative stress. Now, what does that mean? That means that you have a production of these reactive species, these reactive oxygen species called free radicals. And you have one here, such as oxygen free radicals. Now, these have unpaired electrons. Now, this actually isn't oxygen, due to too many electrons, but it's just so you get the idea. But they basically have unpaired electrons, which are extremely reactive and unstable. And it can actually damage the cell from inside by reacting with various different things and lipids, proteins, nucleic acids, whatever it might be. And you don't want to have too many of these reactions because you don't know what could happen, what it could react with. So that's why you have antioxidants, which can donate that electron, or they can degrade it to a form where it can't cause damage anymore. So oxidative stress is basically caused by an imbalance of antioxidants and free radical species. But there's a defense mechanism that most cells, not just cancer cells, but regular cells do when they have oxidative stress or when they have a really high metabolic rate. They need more glutathione. They need more antioxidants. So what they do is glutamate, which is a waste product in the tumor cell from other processes, is in excess. And glutathione production requires a nitrogen source. So the cell leverages that by using this transporter called the XCT, or cysteine glutamate transporter, which transports glutamate outside the cancer cell and brings in cysteine. Cysteine has nitrogen in it, it's an amino acid. It's broken down into cysteine, and then glutamate binds with cysteine to form gamma-glutamylcysteine via this enzyme right here. And you don't need to worry about it too much. And then glycine binds with this substituent to create that antioxidant glutathione. And the final enzyme in this is glutathione synthetase, which is what actually creates the antioxidant. Right? Now, what happens to all this glutamate that's outside the cell now? That's outside in the bone extracellular space. Now, that can cause a lot of different problems, and there's two pathways that we really focus on. So this pathway, as you can see, first we have the osteoclast here and osteoblast. Osteoblast build bone, osteoclast break bone. If you think of them that way, osteoclasts basically secrete acid, which degrades bone. But they use glutamate as a signaling molecule just to control their balance, to make sure that it's basically communication between them, just to keep things in balance, make sure there's not too much absorption, not too much bone growth. But they use only very small concentrations. And then when you get all of this extra glutamate coming in, that you can interfere with that signaling. It's very complicated, but overall you can either get too much bone absorption or too much bone growth. And that goes back to the exercise which we saw at the beginning, which is when you can have these cavities form or excesses of bone. Now, on the other hand, what also happens is that glutamate comes into all the neurons that surround it. So these are all found throughout the bone. And they bind with receptors and excite them, right? Now, if they bind with primary afferent neurons and signal for pain, that's when you can get that sensation of pain that originates from bone metastasis. And all this glutamate can't be broken down by astrocytes, which normally do their job effectively because there's too much of it. So what happens is your body wants to create more astrocytes to help break down more glutamate. But these astrocytes are immature and they actually can't break it down as effectively. So it doesn't really work out, and the immature astrocytes don't really work. So you get continuous signaling, and initially it's acute pain, which is just when glutamate binds and you get the signal going up and you experience pain. But eventually you can get chronic pain through long-term potentiation, which we already discussed. And so you get increased responses to glutamate and just leads to very significant pain. Malignant gliomas, which are cancerous tumors in the central nervous system, which is unrelated to what we just talked about, have a unique behavior that's actually related to this as well. They release tons of waste glutamate, which overly excites the surrounding neurons in a sense, and it's caused excitotoxic damage, which basically means you open all these channels, you have so much calcium influx, and you get tons of enzymes being activated, such as proteases and phospholipases, and all these enzymes can actually damage the cell by doing unwanted things. If you have too much of anything, it can be bad, and that rings the same for cells as well. So what happens is you get so much activation by glutamate that the cells actually die through something called excitotoxicity, and that creates room for the tumor itself to grow. So this is a concept that they're actually exploring, and it might be what's happening for tumors in your bone as well. Something else to note is that metastasis from breast and prostate epithelial cells are actually more painful, or maybe not more painful is the right word, but they're often more effective at establishing themselves in the bone and utilizing this XCT transporter or similar mechanisms. This is actually because they have more effective amino acid transport, probably because breast and prostate, those tissues are more specialized to wanting to bring in nutrients such as amino acids and create proteins. Just think of the breast milk given to a child. The mother is evolutionarily advantageous to give as many nutrients as possible. So it might be a reason why amino acid transport is so effective in these cells. But this is basically why metastasis in bone could be incredibly intense and more painful when it's from breast or prostate cells in origin. There are other modulators in the system. There's in the system as well, such as sulfasalazine or SSZ. Here's just pull it. It's one of the first designer drugs ever made, and it's a joint combination of an antibiotic and an inflammatory medication. And coincidentally, it's also an inhibitor of the XCT, or the cysteine glutamate transporter. Now, it has the ability to increase cellular oxidative stress because it's basically reducing the cell's ability to bring in the nitrogen source that it needs to create glutathione and take care of those oxygen free radicals. But the basis that we're really going to look at it is decreasing pain. By inhibiting glutamate being released, it can affect glutamate signaling to primary afferent neurons. However, you need to note that it does not affect tumor growth, and none of the studies found that it did affect tumor growth. But it did work on pain, and it decreased pain significantly. Now we'll look at some experiments. First, we'll look at the osmotic pump, which is basically this little tablet that can be implanted in mice. And you put a certain drug in it, and it delivers that drug at a continuous and controlled rate over several days or hours at a time. Actually, I mean several days to weeks, actually. Now, what it is, it has this osmotic property where it can take in water and squeeze out the drug very slowly over time. So that way you don't have to keep injecting the mice. Now, why is that beneficial? Well, in a pain experiment, you wouldn't want to keep injecting mice because that can only increase their threshold and can skew your results, especially when you're testing for how much pain they can feel. So this just makes things easier in these types of experiments. Another thing to keep in mind is the dynamic weight bearing device, which you put in the mice and it walks around a bit, and the computer spits out exactly what percentage of their weight is bared on each limb. And as a result, it lets you know whether they're favoring certain limbs over other ones. Another thing to talk about is cancer-induced depression. Now, this is a special type of depression that has a biological basis. So even before people know that they are diagnosed with cancer or they have cancer, they may express symptoms of this type of depression. Now, it's special in that it's not really affected by noradrenaline reuptake inhibitors or serotonin reuptake inhibitors or that sort of thing, or analgesics like anesthesia, stuff like that. However, glutamate drugs do work, like ketamine, which is an NMDA receptor inhibitor that works, as well as sulfasalazine, which is a CTX inhibitor. So basically, going back in this slide, sulfasalazine, which inhibits this transporter right here, reduces the amount of glutamate being released, and that actually has an effect in reducing cancer-induced depression, which is very amazing. But yes, that's about it for pain 2. There's not too much to worry about here, just a few key things to keep in mind. I'll say this diagram is very important, and having a good grasp of this whole system.